Hello, help keep these videos free, current, and available for every student. Consider supporting by scanning the QR code to contribute. Any amount helps. Thank you.
If your project will involve microorganisms, recombinant DNA, fresh or frozen tissues, primary cell lines, established human or primate cell lines, blood products, or other body fluids, you will complete Form 6A. This is your official risk assessment for potentially hazardous biological agents, and it must be approved before you begin any work. It's completed by your qualified scientist or direct supervisor together with you, and every question needs an answer. Attach extra pages if you need more space. Let's start at the very top. You will write your name or team names and the exact project title so it matches your other forms. Right away, remember the spirit of this form. Your study will minimize risk to people, animals, and the environment. That's baked into the 2025-2026 rules. Now you'll move into the first big part, Section 1, which is your project assessment. First, you will identify every agent or material you'll handle. Name the organism or material, include the strain when you know it, say where it comes from, list how much you expect to use, and note its biosafety risk group. For any cell lines or tissues, include the origin or supplier. This tells reviewers exactly what's in play. Next, you will describe the biosafety level of the place you'll work. Say clearly whether the lab is BSL-1 or BSL-2, and make sure that aligns with the organism and the procedures you're planning. If your work will be done in a BSL-2 lab that is not at a regulated research institution RRI, you will include the BSL-2 checklist with your packet. Then you will explain how you'll keep everyone safe. List your personal protective equipment, like gloves, goggles, lab coat, closed-toe shoes, and any engineering controls, such as a biosafety cabinet and its type. Describe safe techniques you'll use, keeping cultures sealed when required, disinfecting benches, handling sharps carefully, and never eating or drinking in the lab. Spell out supervision too, so it's clear who will be present and trained. After that, you will describe how you'll dispose of everything that might be contaminated, plates, tips, cultures, and waste. Match the method to the biosafety level and the agent. Acceptable methods include autoclaving, disinfecting with a proper 10% bleach solution for the correct contact time, institutional biohazard pickup, or other manufacturer-approved methods. The rules give these examples, and your disposal plan should follow SDS guidance and lab policy. Now comes Section 2, Training. You will list the training you'll receive, standard microbiological practices, BSL-1 or BSL-2 procedures, bloodborne pathogen training if you'll handle body fluids, and any instrument training such as incubator or biosafety cabinet use. Your direct supervisor will also note their own relevant training and experience. This section shows you'll be prepared and properly supervised. Next is Section 3, the set of checkboxes that clarify where the work will happen and what approvals apply. If your work will not be at a regulated research institution, your qualified scientist or direct supervisor will check whether it's a BSL-1 or BSL-2 lab. Remember that BSL-2 work outside an institution needs that BSL-2 checklist attached and SRC approval before you begin. If your project will culture multi-drug resistant organisms, you will check the MDRO box. These studies are conducted at an RRI at minimum BSL-2 with Institutional Biosafety Committee IBC pre-approval, and you'll record the IBC approval date. If your study will be conducted at an RRI, you will attach the institution's IBC or Institutional Animal Care and Use Committee IACUC approvals as applicable. Note the origin of any cell lines and include the approval date. If your RRI doesn't require IBC or IACUC for your specific work, that option is also indicated. Check the boxes that truly match your situation and attach the proof. To finish the form, your qualified scientist or direct supervisor will certify the assessment. They will confirm they reviewed your plan and documentation, that the information is accurate, and that the study is approved at BSL-1 or BSL-2 and will be conducted in an appropriate lab. They will print their name, sign, and date the review before any experimentation begins. This signature is your green light to proceed safely and in compliance. A few quick guardrails from the rules while you're completing 6A. Home culturing of potentially hazardous agents is not allowed. You'll work in a proper lab with the right containment and supervision. Anything above BSL-2 is off-limits for pre-college research. Unknown cultures that stay sealed can be treated as BSL-1, but the moment a container is opened, it becomes BSL-2 with BSL-2 precautions. And at the end of the study, disposal must match the organism and biosafety level, autoclave, disinfect correctly, or use approved biohazard pickup. One last planning tip. If anything in your plan changes, organisms, methods, location, or disposal, you will pause and seek reapproval. Any proposed change after initial approval must be reviewed again by the SRC, IBC, or IECUC before you resume. That's how you keep your work aligned with the rules from start to finish. Complete each prompt clearly, attach any required institutional approvals or the BSL2 checklist when needed, and make sure the certification is signed and dated. With a thorough, accurate 6A, you'll show reviewers that your study will be safe, compliant, and ready to begin.
Remember, help keep these resources free, current, and available for every student. Consider supporting by scanning the QR code to contribute. Any amount helps. Thank you.